Welcome to this presentation on organisms exchange substances with their environment. All living organisms need to exchange substances with their environment to survive. They take in useful materials such as oxygen, water, and nutrients, and remove waste products such as carbon dioxide and urea. The efficiency of exchange depends on factors such as surface area, concentration gradients, and the organism size. Specification, context, and importance of exchange. Why organisms need to exchange substances. Organisms exchange substances with their environment. Underpins understanding of how living systems survive, grow, and maintain internal balance. Exchange refers to the movement of substances such as oxygen, carbon dioxide, nutrients, and waste products between an organism and its surroundings. This topic integrates principles of diffusion, osmosis, and active transport with large-scale biological systems including gas exchange organs, the digestive system, and mass transport systems in animals and plants. One key reason this topic is essential is that it links molecular-level processes, such as diffusion across membranes, with whole organism adaptations like lungs, gills, and vascular tissues. For example, oxygen must diffuse across the alveolar membrane in humans before being transported by hemoglobin to respiring tissues. Another important aspect is the relationship between size and exchange. As organisms increase in size, their surface area to volume ratio decreases, meaning diffusion alone becomes insufficient. This drives the evolution of specialized exchange surfaces and transport systems, a concept that recurs throughout the specification. All living organisms must exchange substances with their environment in order to survive, and this requirement becomes more complex as organisms become larger and more metabolically active. Cells need a constant supply of oxygen for aerobic respiration, which releases energy in the form of ATP. They also require nutrients such as glucose, amino acids, fatty acids, water, and mineral ions to build macromolecules and maintain cellular processes. At the same time, waste products such as carbon dioxide, urea, and excess ions must be removed to prevent toxic effects. In unicellular organisms like amoeba, exchange occurs directly across the cell surface membrane by diffusion and osmosis because every part of the organism is close to the external environment. However, in multicellular organisms, most cells are far away from the body surface, so simple diffusion alone would be too slow to meet metabolic demands. This is particularly important in active organisms such as mammals, where tissues like muscles have high oxygen demands. As a result, specialized exchange surfaces and transport systems have evolved. Another key concept is the maintenance of diffusion gradients. Exchange is only effective if there is a steep concentration gradient, meaning substances must be continually supplied or removed. For example, ventilation of the lungs and blood flow through capillaries maintain low oxygen concentration in the blood at the alveoli, allowing oxygen to diffuse rapidly. In plants, the uptake of carbon dioxide for photosynthesis and the loss of oxygen and water vapor are balanced through stomata. Surface area to volume ratio and its biological significance, general features of efficient exchange surfaces. Surface area to volume ratio, SAV, is a fundamental concept in understanding why organisms need specialized exchange surfaces. As an organism increases in size, its volume increases faster than its surface area. This means that for larger organisms, there is less surface area available per unit volume for exchange to occur. For example, a small organism such as a flatworm has a high SAV ratio, allowing it to rely on diffusion across its body surface. In contrast, mammals have a very low SAV ratio and therefore require lungs, a circulatory system, and ventilation to meet metabolic demands. This concept is often assessed through data interpretation questions, where students are asked to explain trends or predict outcomes. SAV ratio also explains why cells are small and why structures such as microvilli in the ileum or root hair cells in plants are highly folded. These adaptations increase surface area without significantly increasing volume. Efficient exchange surfaces share a set of common features that maximize the rate of diffusion. One key feature is a large surface area, which increases the amount of substance that can diffuse at any one time. Examples include the alveoli in the lungs, villi in the small intestine, and root hair cells in plants. Another essential feature is a thin exchange barrier, which shortens the diffusion pathway and increases the rate of diffusion according to Fick's law. In alveoli, the barrier is only 2 cells thick, the alveolar epithelium and the capillary endothelium. Exchange surfaces are also often moist, allowing gases to dissolve before diffusing across membranes. Additionally, efficient exchange systems maintain steep concentration gradients. This is achieved through mechanisms such as ventilation in lungs, mass transport systems in animals, and continuous water flow in fish gills. A rich blood supply or transport medium is another common adaptation, ensuring rapid removal or delivery of substances. Gas exchange in insects, the tracheal system, gas exchange in fish, countercurrent flow in gills, gas exchange in plants, stomata and guard cells. Insects rely on a tracheal system for gas exchange, which is well adapted to their relatively small size and high metabolic activity. Spiracles are openings along the sides of the insect's body that can open and close to control gas exchange and reduce water loss. Air enters through the spiracles and travels along the tracheae, which are supported by chitin to prevent collapse. These branch into very fine tracheoles that extend directly to individual cells. This arrangement means that oxygen is delivered straight to respiring tissues, reducing the need for a circulatory system to transport gases. Diffusion is the main mechanism of gas exchange at the tracheoles, but in active insects, abdominal movements can ventilate the system, maintaining steep concentration gradients. One limitation of the tracheal system is that diffusion becomes less effective over large distances, which helps explain why insects are relatively small compared to vertebrates. Fish exchange gases using gills, which are highly specialized organs adapted for extracting oxygen from water, where oxygen concentrations are much lower than in air. Gills consist of gill filaments, each covered in many gill lamellae, providing a very large surface area. Blood flows through the lamellae in the opposite direction to the flow of water over them. This countercurrent arrangement ensures that blood with a lower oxygen concentration always meets water with a higher oxygen concentration, maintaining a diffusion gradient along the entire length of the lamella. As a result, fish can extract up to 80% of the oxygen from water, compared to much lower efficiencies if concurrent flow were used. Gills also have a thin exchange surface and a rich blood supply, both of which increase diffusion rates. However, gills are inefficient in air because the filaments collapse, reducing surface area. Plants exchange gases with their environment primarily through structures called stomata, which are pores found mainly on the underside of leaves. Stomata allow carbon dioxide to diffuse into the leaf for photosynthesis, while oxygen, a waste product of photosynthesis, diffuses out. Each stoma is surrounded by two guard cells that control its opening and closing. Guard cells have unevenly thickened cell walls and respond to changes in turgor pressure caused by the movement of potassium ions and water. When guard cells are turgid, the stoma opens. When they are flaccid, it closes. This mechanism allows plants to balance the need for gas exchange with the risk of excessive water loss. Inside the leaf, large intercellular air spaces increase surface area and allow rapid diffusion of gases to photosynthesizing cells. Unlike animals, plants do not actively ventilate their exchange surfaces, relying instead on diffusion driven by concentration gradients. The human gas exchange system and ventilation, alveoli as highly specialized exchange surfaces. The human gas exchange system is adapted to meet the high oxygen demands of a large, active organism. This system includes the trachea, bronchi, bronchioles, and alveoli. Air is ventilated into the lungs through pressure changes generated by the diaphragm and intercostal muscles. During inspiration, the diaphragm contracts and flattens while the external intercostal muscles contract, increasing thoracic volume and decreasing air pressure inside the lungs. This causes air to move into the lungs. Expiration is usually passive, involving relaxation of these muscles and elastic recoil of the lungs. Ventilation plays a critical role in maintaining steep diffusion gradients for oxygen and carbon dioxide at the alveoli. The branching structure of the airways ensures that air is delivered efficiently to millions of alveoli. Alveoli are the primary site of gas exchange in the human lungs. Humans have around 300 million alveoli, giving a total surface area of approximately 70 square meters. Each alveolus has a very thin wall, composed of a single layer of squamous epithelial cells, which reduces diffusion distance. Alveoli are also surrounded by a dense network of capillaries, ensuring a rich blood supply that rapidly transports oxygen away and brings carbon dioxide to be removed. The alveolar surface is moist, allowing gases to dissolve before diffusing across membranes. Ventilation and blood flow together maintain steep concentration gradients for both oxygen and carbon dioxide. In addition, alveoli contain elastic fibers that help them inflate and deflate during breathing. Damage to alveoli, such as an emphysema caused by smoking, reduces surface area and impairs gas exchange. Digestion and absorption in the small intestine, the mammalian heart and double circulatory system, blood vessels and transport of substances, transport in plants, xylem and phloem, required practicals, exam skills, and key summary. Digestion involves the breakdown of large, insoluble molecules into smaller, soluble ones that can be absorbed into the bloodstream. This occurs through mechanical and chemical digestion, with enzymes such as amylase, protease, and lipase playing key roles. Absorption takes place mainly in the ileum of the small intestine, which is highly adapted for this function. The ileum is long, increasing the time available for absorption, and its lining is covered in villi and microvilli, greatly increasing surface area. Each villus contains a rich network of capillaries and a lacteal, allowing efficient transport of glucose, amino acids, fatty acids, and glycerol. The epithelial cells have many mitochondria, providing ATP for active transport where required. Mass transport in mammals relies on a double circulatory system, consisting of the pulmonary and systemic circuits. The heart is a muscular pump divided into 4 chambers, 2 atria and 2 ventricles. The right side of the heart pumps deoxygenated blood to the lungs, while the left side pumps oxygenated blood to the rest of the body. This separation prevents mixing of oxygenated and deoxygenated blood and allows higher pressure in the systemic circulation, ensuring rapid delivery of oxygen and nutrients. The cardiac cycle consists of atrial systole, ventricular systole, and diastole, with valves ensuring one-way flow of blood. Understanding pressure changes and valve function is essential for explaining heart action in exams. The double circulatory system is an adaptation to high metabolic demand and links directly to efficient exchange at tissues. Different types of blood vessels are adapted for specific roles in mass transport. Arteries carry blood away from the heart at high pressure and therefore have thick muscular walls, elastic fibers, and a narrow lumen. Veins return blood to the heart at lower pressure and have thinner walls, a wider lumen, and valves to prevent backflow. Capillaries are the sites of exchange between the blood and tissues. They have very thin walls, only 1 cell thick, and a small diameter, which slows blood flow and allows efficient diffusion of substances such as oxygen, glucose, and carbon dioxide. The extensive capillary network ensures that every cell is close to a blood supply, reducing diffusion distance. Understanding these structural differences allows students to explain how substances are transported efficiently around the body. Plants also require mass transport systems to move substances over long distances. Xylem transports water and mineral ions from the roots to the leaves in a process driven by transpiration. Xylem vessels are dead, hollow cells with thick lignified walls, providing strength and preventing collapse under tension. The cohesion tension theory explains how water is pulled up the plant as a continuous column due to evaporation at the leaves. Phloem transports organic substances such as sucrose and amino acids in a process known as translocation. This occurs by mass flow, driven by pressure gradients between sources, such as leaves, and sinks, such as roots or growing tissues. Phloem tissue consists of sieve tube elements and companion cells, which are adapted for active loading and unloading of sugars. Practical involves investigating the effect of surface area to volume ratio on the rate of diffusion using agar cubes and acid. This practical reinforces theoretical understanding and develops skills in data collection, analysis, and evaluation. Students must be able to identify variables, assess limitations, and suggest improvements. In this topic, we have explored how organisms exchange substances with their environment. Efficient exchange depends on specialized surfaces, large surface areas, short diffusion distances, and transport systems that move substances around the organism. Thank you for watching.